Good morning, dear students. We welcome you for the today's e-tutorial on a type study of Funaria, where we will be dealing with the morphology, reproduction, and life cycle. This is Dr. M. Padmasundari, Assistant Professor, Department of Botany, St. Joseph College for Women. In this particular type study, we will be discussing about the classification of Funaria, external morphology, the types of the reproduction, and the sporophytic phases of the Funaria. So when you see the classification, the the Funaria belongs to class Bryopsida. The order is Funarias, family is Funariaceae, and genus is Funaria. This is the most common moss found throughout the world. 120 species have been recorded in the world, and India 15 species have been reported. They grow on moist grounds, in closed tubs, and on damp and shady moist rocks, cervices, tree trunks, and other similar surroundings. Funaria hygrometrica is a cosmopolitan and best known among the Funaria species. If you see the external morphology, the Funaria is divided into two stages: the leafy gametophyte, and the sporophyte. The gametophyte is the main plant body and is presented by a juvenile and adult stage. The haplite plant body is divided into the haplite plant body is divided into root, stem, and the leaf. The stem is erect, branched, green, and photosynthetic. The branches arise from the lower portion of the stem. The leaves are arranged spirally. Mm, uh, uh, on the stem. They are scattered towards the base and crowded at the apex. They are sessile and almost ovate in shape and with a distinct midrib. The stem is fixed to the substratum by rhizoid. The stem is fixed by the substratum by the rhizoids. The rhizoids are branched, multicellular, and appears like root hairs. The gametophyte plants are monoecious and homothallic. The male reproductive organs and the female reproductive organs are born on different branches of the uh, same plant. Both antheridia and archegonia are produced in clusters at the end of antheridia and archegonial branches, respectively, and the sporophyte is attached to the archegonium in parasitic nature and divided into foot, seta, and capsule. So this is the sporophyte where you can see the sporophyte, and this is the gametophytic plant where the rhizoids are septate. This is the leaf structure with a ovate and a midrib present in that. If you look into the anatomy of the stem, the anatomy consists mostly, it is divided into the epidermis, the cortex, and the central stand. Epidermis is the outermost, made up of cells with chloroplast. Cortex is well parenchymatous region, axis just inner to the epidermis, and is extended up to the central stand. And they consist of chloroplast when young, but disappears when they mature. Central in the center is present the central strand, which is narrow, colorless, thin-walled cell and lack protoplasm and thus dead cells. They help in the conduction of water and the other nutrients. Regarding the anatomy of the leaf is very simple. The lamina consists of a single layer of the large thin-walled cells. These are the thin-walled cells with many chloroplasts and serves as the major photosynthetic regions. In the center is the lamina is present a midrib and made up of small strands of narrow thick cells which helps in the conduction. Regarding the reproduction in Funaria, it takes place by vegetative and sexual methods. Vegetative methods includes by primary protonema, secondary protonema, bulbils, gemme, and epospore. Protonema, here the spore germinate to produce branch filamentous multicellular structures called primary protonema. These break into filaments to produce new protonemal cells which develop into a leafy gametes. The secondary protonema is developed from the germination of the spores and is called secondary protonema. It is developed from the broken or injured rhizoids, stems or leaves and give rise to whole new gametophyte. Bulbils are the resting bodies that arise from the rhizoids and they divide in favorable conditions and produce the filamentous protonema which grow into a leafy gametophyte. The fourth type of the methodology is known as the gamete. These are in unfavorable conditions, the terminal cells of the protonema divide transversely and longitudinally to give a 10 to 30 cell green bodies called gamete. These structures starts to divide and form new plants in favorable conditions. Apospore is a development of gametophyte from the sporophyte without the spore formation. Vegetative cells of the sporophyte give rise to green protonemal fragments which later become the gametophyte. The gametophyte produced from apospore is diploid and gives rise to tetraploid and the tetrasporophyte. So these are the different methods of the vegetative reproduction where the protonema, the B is the protonema. So this is the structure by which the reproduction takes place. Reproduction, sexual reproduction, if you see in Funaria, it is a gamous type where the species are monoecious, thereby saying the male and female are developed on the different branches of the same plant. The male cell organs are antheridium. They are the branch is about 1 centimeter in depth. The branch bears reddish brown to orange antheridium of different stages of development. The antheria is surrounded by a rosette of leaves called perigonial leaves. The antheridial clusters along with the leaf of rosette is known as perigonium. Numerous antheridias are found and are known numerous uh, are found in the antheridia. The hair-like structures are found and they are known as the paraphyses. The store water with the help in photosynthetic of dehiscence of antheridia and protect the growing male reproductive organs. So this is the antheridia which is found on the antheridial branches which consist of the sterile leaves, the paraphyses and these are the antheridia having the sterile jacket cell and these are the antheridial spores which on germinating develops the androcytes. The archegonial branch develops laterally at the base of the male branch. At the apex of these branches grow archegonia, surrounded by pericytial leaves and paraphyses. Mature archegonium consists of stalk, a swollen venter, and an elongated neck, and bilayered archegonial jacket surrounds the venter region, while in the neck region it is single layer. The neck continues in a rows of 6 to 9 neck canal cells, and the venter contain a venter canal cell and an egg cell. This is the structure where on the ventral surface they have a pericytial leaves, the paraphyses, and these are the archegonia or the female gametophyte consisting of the neck canal cells, the neck, the ventral canal cell, and the eggs. Egg. So fertilization is facilitated by rain or dewdrops. The biflagellated anthrozoids are set free during the process of dehiscence of antheridium. At the same time, the neck canal cells and the venter canal cells of the archegonium disintegrate and degenerate to form the mucilaginous mass. Anthrozoids carried by the raindrops fertilize the egg and form the zygote, the first cell of the sporophytic phase. The sporophyte is now divided into three parts, the foot, seta, and the capsule. The foot is the swollen part of the sporophyte and remains embedded in the tips of the female branch. It is composed of parenchymal cells. The seta is a long reddish brown cylindrical structure, connects the capsule to the foot, and it is composed of outer epidermis, middle cortex, and a central cylinder. The cortex is made up of thick walled cells, and a central cylinder is made up of elongated thin walled cells, and they functions to carry food and water from the foot to the capsule. Capsule is the body of the sporophyte where the spores are developed. It is green, yellow, or orange in color and is pear shaped. The capsule consists of three parts. Those are known as apophysis, theca, and lid. Apophysis is the basal sterile part of the capsule that is a direct contact with the seta. The theca is the middle portion of the capsule made up of central sterile core called the columella, and the lid is the operculum in the upper terminal portion of the capsule. So this lid is separated from the theca by a narrow circular constriction. Just above the constriction is 5 to 6 thin walled cells called annulus. Below the walls of the lid are two ring structures, the peristome teeth. The outer ring of the peristome teeth is exostome and the inner ring is known as the endostome, which takes place in the dehiscence of the capsule. So this is the structure of the capsule where this is the steta, this is the apophysis, the middle part, the columella, we call it as then the air cavities are present, and here is the what you call the center part is known as the columella, and this is the spore sac inside which the spores are present, and these are being dispersed by the peristomial teeth which are present here on the upper with the annulus like the structures. As the capsule matures, the operculum is thrown off by the rupture of annulus, exposing the peristome teeth to the air. The capsule dries slowly and the peristome teeth ruptures, and they form a fringe around the mouth of the spore sac and releasing the spores in a small amount. So this is how the spores are being released off from the peristomial teeth. Spores produced by sporophyte are called neospores. They consist of outer layer called the exine and inner layer called the intine. They absorb moisture, swell subs, and rupturing the exine. Intines grows into a germ tube and later into branch filament structure called the protonema. The protenome grows on the substratum by fixing itself with rhizome, and many lateral buds are produced from the growth of protein and give rise to the gametophyte. This is the spore, the exine broken, the intine come in the form of germ tube and they form the structure. The protonema-like structures are being developed. If you see the life cycle of Funaria, it is a heteromorphic type of the life cycle where there is a where the dominant phase is the gametophyte phase and the sporophyte phase is also present. And this gametophyte usually develops. Gametophyte consists of the male and the female branches on which are present the archegonium and the antheridium. The antheridium later at the time of maturity produce the anthrozoids. These anthrozoids with the raindrops or dewdrops, they fall on the mucilaginous of the neck cells of the archegonia where they fuse and form the and the fusion of the gametes and formation of the zygote is the first oospore is the first sporophytic phase. Later it divides to two cells zygote and it forms the epibasal and the hypobasal, and these two cells later further divides and they form the they form the sporophyte which is dependent on the archegonian female gametophyte, and that is divided into foot, seta and the capsule. So foot gives support, this is the foot and the is the anchorage to the sporophytic plant, and the seta helps in the absorption of the nutrients, and the capsule mostly present the spores where they disperse for mother cell. It undergoes reduction division, and this is again the beginning of the gametophytic phase for the formation of the spore tetrads. The spore separates. The spore is a double layer structure consisting of exine and intine. When the exine ruptures, again the intine, the germ tubes will be developing it. So in this way, the Funaria shows the heteromorphic type of the life cycle, showing a gametophytic dominant phase and a sporophytic parasitic phase on the gametophytic plant architecture. So these are the questions related to the the morphology and the life cycle of the Funaria. Where the questions are: is the plant body of Mosvena gametophyte or a sporophyte? It is a gametophyte. What kind of flagellations do anthrozoids of Funaria have? So they have flagellation. And anthrozoids have biflagellated structure. How does vegetative reproduction in Funaria takes place? So they takes place by means of the primary secondary protonema or spore. So there are few of the things that takes place. So thank you for your listening, and any queries please inquire.